like Sean said, my name is Duke Pauli. I'm a graduate student at Montana State University, and I work on barley breeding. So today we'll talk about GAPIT and using that for genome-wide association studies. And I'm sure most of you are familiar with GWAS already, and some of the packages that are out there that are currently available. GAPIT is just another one that you may choose to use. It has a lot of functional utility, and I personally like it a lot. And hopefully by the end of today's webinar, you will too. So here's just a brief overview for what we'll be covering today. I've kind of broken it down into 4 parts. Part 1 is kind of an introduction, some background information, as well as installing RStudio. I will be doing my R demo in RStudio. It's a pretty nice user interface that makes R just a little bit more manageable. Part 2 will be loading some sample files, going over the sample files, loading GAPIT source code so you can actually initialize the program. And then part 3 will be doing the loading of the files, checking the data, and then doing the actual analysis. And then part 4 we will cover the output that GAPIT generates. And I'll take some questions, and then I actually have a live demo that I'd like to run at the end. So some of the learning objectives for today, just briefly installing RStudio. It's pretty straightforward. Sean asked that I cover it just so people get a little bit more exposure to it. Also, we'll be loading the packages and the source code for GAPIT, sample files, basic analysis, and again covering the output so that you guys can get the most out of using GAPIT. Just as a brief disclaimer, what I'm assuming for today's tutorial is that you are familiar with R and some of the basic commands like loading data, how to type commands, kind of the structure of it. And I'm also kind of assuming that you have an understanding of the principles of GWAS. Obviously there's a lot of theory behind it, there's a lot of statistical knowledge behind it that I will not be covering today because that could cover an entire semester. So again, just to clarify, what I won't be covering is installing and running R. There's a great tutorial on the extension website that covers this and gives a very good introduction to the program. And also, I won't be covering GWAS and the theory behind it. And finally, the genomic prediction functions of GAPIT. I won't be going over that either. It is capable of doing that, but for the sake of time, I can only cover so much. So what is GAPIT? From the name, it's Genome Association and Prediction Integrated Tool. It's a statistical package that was developed by Alex Lipka and Ziwu Zhang at Cornell, and they are members of Ed Buckler's group. And to, I guess, be somewhat coarse, it's kind of like Tassel, but it's implemented in the R software environment. And I think that offers a few advantages that maybe you don't get with Tassel. And it's perhaps one of the reasons that I think I like using it, and I think why some other people are enjoying the benefits of using it. And I know this paper is posted on the website, but I did just want to throw in the citation in case you didn't see it. So you might be asking yourself, why should I be using GAPIT? There are already a bunch of programs out there that do GWAS, but one of the things that's becoming an issue as the genomic era progresses is the size of the data sets, and they are getting quite large. I don't even work with a very big one. I think it's probably only like 15 million cells for the genotype data, and so I've already seen performance problems, and so GAPIT has a unique ability, again, because in R, it has some options available to it like file splitting, and that allows you to split up your data into multiple files so you can bring it into R in separate pieces and then kind of run your analyses once it's in the environment. Some of these other programs have complex user interfaces, some are even command line driven. So the question I guess maybe you have to ask yourself is, how willing are you to learn another program? And most of you are already familiar with R, so it just makes sense to kind of just stick with one. And I think a lot of the time people think maybe because it's an R or some kind of command line program that it requires a lot of code. And as you'll see today, it does not. It's actually pretty straightforward. Again, some of the reasons I like it are the data manipulations, analyzing field data, calculating various statistics. You can all do that in one program. You don't have to do, adjusted means in SAS and then import that in a tassel. So it's very nice. So supporting information is excellent. It's just outstanding. They have a wonderful user manual, and it's one of the unique features is GAPIT was designed so that you can take their user manual and essentially cut and paste their code out of the manual and put it into R and run it. It is easy to use. The developers, especially Alex Lipka, extremely helpful. He has so many dumb questions that I've had and has always been very gracious about it. And then finally, from a performance issue, I've found that GAPIT is quite fast. Some of the analyses I ran in Tassel used to take me a better part of a day and a half. And when I switched to GAPIT, I was able to do them in 3 to 4 hours. So, I mean, your time is valuable, so that's, a consideration. Installing RStudio, just briefly, you can go to the website listed here, select Download Now, and download for desktop. And it'll essentially walk you through the installation of RStudio. It's pretty easy. And RStudio is a very nice environment to work in. You'll see in this photo here. Right here you have your script window, and then down here is the actual R console. Over here is your workspace image. So in this case you can see that I have data loaded, I have all the GAPIT functions loaded, and then down here is kind of where you have your plots. You can also manage your packages, and at the demo at the end we can explore some of those utilities. So running GAPIT, my biggest piece of advice would just be use the manual. It's like an R vignette, but it is literally 1,000 times better. It has all the code. You can copy and paste it like I said. They outline several scenarios that you might encounter with your own data, and one of the more important things is how it describes how to format the data for input. As just about everybody knows, this is where most of your time is spent. Doing the actual analyses doesn't actually take that long. And then again, it lists all the options for doing the analyses. Today I will present but a short list of them. There's a lot of them, and that's kind of the power of GAPIT, is how much stuff that you can do. It also describes the output that's generated. Because it's in R, it has the ability to create a lot of graphs. I guess I shouldn't speak out of turn. I'm not sure if the new version of Tassel is doing this, but it generates a lot of the output that you want, like Manhattan plots, QQ plots, allelic effect estimates. And then finally, the manual, kind of towards the end, explains some more advanced features of the program, and we might touch on those in the demo as well. So I've included a few sample files on the website. These were all generated from the barley cap, which is similar, you may have like the soul cap and whatnot. The data is available from this website, the Trudi ACA Toolbox, and the one that we'll be analyzing today is data from a replicated yield trial. There's 768 lines, and that is comprised of 8 separate breeding programs, each submitting 96 plus 10 check lines. And the trait that we'll be looking at is grain protein content. This trait is extremely important in terms of malting and brewing. If your beer has too much protein in it, it gets hazy and actually starts to precipitate out. And just to clarify, GAPIT does handle multiple traits per run. So the sample files, if you have time to look at them, you'll notice the genotype data is in the HAMPMap format. This is becoming quite common format for the data. But again, check the manual because there's a lot of other options. And so the 2 files that are up are the genotypes in the HAMPMap format, and then the protein content of the grain, which is in percentage. And the markers, the number of markers we're using is 2,859, and they've been filtered on missing data set at 20% and a minor allele frequency of 1%. For those of you not familiar with the HAMPMap format, it's pretty much a genetic map and marker data put into one file. When you bring it into R, I know for me personally where I get my download of my data, they include some pound signs, and it's important to make sure those aren't in the headers. They typically show up in the assembly number column. And then it's kind of unique about GAPIT, it doesn't actually use all the HAMPMap information. Really what it cares about is the name of the SNP, the chromosome, and the position. Everything else can be filled in with NAs with regard to like the assembly, the center, and some of the other information. And when you do the actual import, you want to make sure that you set the header equal false command. And so to give you a better idea of this, here's RS. So in the barley cap data, our markers are denoted by the actual BOPA, or the genotyping chip. And so here you can see the name of the marker, the alleles, whether it's AG, what type of SNP it is, excuse me, the chromosome, the position, and this strand through Q code. This is the stuff I was talking about that you can leave as NA. It doesn't screw up GAPIT, and GAPIT doesn't really need that information. And then finally over here, you can see these are the actual lines, Harrington robust, and you can see the SNP genotype call. So this is what HAPMap data looks like. I know at this point some of you might be saying, well, my data is not in HAPMap format. Not to worry, GAPIT handles lots of stuff, and we will talk about that in a little bit. The phenotype file is pretty much just a straightforward text file. Use taxa as the header for the entries, the lines that you're evaluating. And like I said, GAPIT handles multiple phenotypes. It's just that when you submit the phenotype file to the program, you need to have each trait in its own column. And then missing data should be coded as NA or NAN. And when you import the data, use header equal true. And so here's just kind of a quick image of what the file looks like that we'll be looking at today. And like I said, like if you had another trait like perhaps heading date or plant height, you would just enter heading date here and have the subsequent entries for that for each line as well as plant height. And then when GAPIT runs the analyses, it sees that there's multiple columns, and so it automatically does the GWAS for each trait. So there's no extra programming required on your part. So in terms of running GAPIT, it needs some other programs. GAPIT uses source code from the Buckler website, which is the same spot where you can get Tassel. And they use source code because it allows them to update the package continuously without always having to download a new version from the crayon mirror that you typically have to do in R. So that's kind of beneficial and helpful. You'll notice at the bottom it says, important if GAPIT isn't loading, check the website. Sometimes they update the code in the packages that you need to run it, and they'll typically tell you. So GAPIT also uses some of the bioconductor packages, as well as some other ones that have been generated, and then it also uses EMMA, which is a program for estimating variance components in the P plus K model. And then finally, there's the functions install packages and library, and I'm sure most of you are familiar with that, but we can cover that in the demo. And so these are what the first few lines of code actually look like. So here's the bioconductor code, the install, you need the gplots, LD heat map, and genetics, and then the library will actually load them. And then down here you can see the source code, and you can see that GAPIT functions, that's, tells R what function it's going to get, and then finally here's EMMA at the bottom down here. So uploading files, most of you are probably already familiar. In RStudio, you can do it just by point and click. It's under the Tools tab, and then Import Dataset, and then it has like a little wizard that will walk you through it. Manually, which you probably already use, which is if you know the address of the file that you need. And then finally, there's a piece of code called file.choose, which opens a file browser, and then that allows you to navigate through your directory to find the file that you want. And I work with all my data in a text file, and so I always use the command read.table, and there is an error. That should just be a colon, not a semicolon. So down here, in the R code that I've given you, there's a lot of annotation. I hope it hopefully explains everything pretty explicitly. Here's this piece of code file.choose, and then finally, here's the actual importing of the data. Again, remember that header needs to equal false for the HAPMap genotype file format. And then finally, the phenotype file here at the bottom. So doing analyses, I think the biggest piece of advice would be knowing your data, knowing what it looks like, knowing how it was generated, what your family structure is like. I got one question about analyzing a NAM population with GAPIT. And the short answer is, yeah, you could do a preliminary analysis using GAPIT, but you probably want to do something else given the unique population structure behind the NAM population. So again, just know your data, and then do a quick check, to make sure everything's properly loaded. Plot the data, look at the descriptive statistics, just to make sure everything's looking right before you actually run the analysis. And then I'm just giving you some basic code to help you do that. Structure, it shows you the structure of the data frame, obviously a histogram, and then I just calculate some basic summary statistics and this line of code down here to make sure, either A, that I know that there's missing lines, so that way if something didn't get loaded right, it'll become pretty apparent. And then so running that code, here is the output of the structure command. It tells you, it's a data frame. There's 768 observations of 2 variables, taxon, protein. Gives you that information. And then finally over here in RStudio, there's this continuous plot window. And here's just a plot of the histogram of the data. And it looks, pretty normally distributed. We have a few outliers perhaps in the tails, but given the sample size, it's kind of what we would expect. And then finally, again, you can see up here are the actual data files, and then here's the GAPIT function. That's a bit important in the workspace. So to run GAPIT with the HAMPMap formatted data, we need 2 files, the genotype file and just the phenotype file. In the actual code, there's a bunch of parameters, and again, refer to the manual. There's more than I can cover today. And so today we'll just do a basic analysis, accounting for population structure, using compression and not using compression, and P3D. Compression and P3D are kind of some more advanced topics that you may be familiar with already. Compression is basically the option of reducing your kinship matrix down to more manageable size, because one of the largest, or I guess most computationally intensive steps is the inversion of a matrix. And so if you can make it smaller, it kind of speeds it up. P3D stands for population parameters previously determined, and basically what GAPIT and then finally, one advantage of the HAMPMap formatted data is that GAPIT can actually handle the imputation of missing marker data. And here are 3 commands based on how you want to do the imputation. So here's just kind of the analysis. Remember that R is case sensitive, so it needs to be capital Y, G. So here we have the method of imputation, the number of PCAs we want to use. This major allele 0, again, reports the allele effect with respect to the minor allele. That'll become important when you look at your minor allele effect estimates. And these 3 lines here, this actually turns off compression. And it just basically says treat each line as its own individual line and don't try and group them. And then finally, make sure you set the working directory where you want the files to be saved. There is a lot of output that is generated. And so there's a few ways you can do it in RStudio. Use the session tab. In R, you can use file, change directory, and then manually with set working directory. And here's kind of what that looks like in the actual RStudio environment. So you can see it's again the same lines of code. And this is when you're not using compression. And this bottom one is if you did. Compression is a default setting in GAPIT, and it does the groupings by 10. And so that's kind of one of the things that you'll have to play around with when you analyze your own data. Here is just a page out of the manual, and you can see, and this is not even close to all of them, just the options that GAPIT has available to it, and that you need to think about and consider when you're doing your actual analyses. And so some of the output that GAPIT generates, you'll see a QQ plot, PCA graph, allelic effect estimates, BLUPs, GWAS results. So there's a lot more information than just this, but this is what I'm going to cover because this is kind of the basic stuff that you need to look at. So again, the QQ plot, it's pretty nice, again, because it's an R. It has really good graphing features. And so it provides a shaded area of what you'd expect for deviation from this fitted line of expected versus observed. And you can tell in our data set we're getting a little bit of deviation from this fitted line. Not totally unexpected. It's a pretty diverse population, and we kind of expect to see this. And then finally, here are the SNPs that are probably significant. Here is the PCA plot. And so in barley, there's two main types. There's 2-row and there's 6-row. And that has to do with how many rows of kernels are on the head. And doing the PCA analysis, things break out into 2 and 6-row. Here's the 6-row germplasm. GAPIT also just a discrete plot. Yeah. And so, you need to use that information to assess how many principal components you might need to use in your model. I chose 3. It's kind of at the bottom of the boot, and that's kind of what people recommend is when it starts to level out. And so again, GAPIT generates all this. It's in a PDF file. Here's what you're probably most interested in. These are the GWAS results. It lists the SNP, the chromosome position. It gives you a raw, uncorrected p-value. And I guess caution about whether or not you want to use that as your significant value. The nice thing about GAPIT is it also gives you the FDR adjusted p-value, and the FDR is based on Benjamin and Hochberg and the method they use. GAPIT also reports the minor allele frequency. And one of the options that you can use is you can filter the results based on minor allele frequency. And so you can set it like 5%. So you'd see, that these lines would be removed, or these results. It also gives you the number of observations that were used, and then the R-square of the model without the SNP and with the SNP. So obviously the difference between these two values is the R-squared of the SNP. And in some cases it would be the R-squared of the QTL. So here's the allelic effect estimates. In that last page you might have saw this marker came up significant. And this just gives you an estimate of the effect. And in our case, the minor allele would reduce grain protein content by about 0.23%. And so again, GAPIT generates this table for you as well. It does do the genome-wide Manhattan plot, and you can set the significance level. And so again, the markers that show up significant for this trait, they're on chromosome 6 and 7. And so you can clearly see that in the diagram. GAPIT also generates, though, chromosome-specific Manhattan plots. So for barley, there's this gene on chromosome 6 that has a very large impact. And there's actually 2 markers. They're just stacked right on top of each other. And so again, those are showing up, and you can cruise through each of the other chromosomes. And this might be a little confusing for some of you if you're not really familiar with compression. Basically, it gives you the methods that it's using. So the optimal method to calculate group kinship, they use the mean. And then there's different clustering methods, and so the best clustering method was average. It tells you that the number of groups was 758, and if you remember, we only had 768 lines, so it treated most of them as individuals. And then finally it gives you, the negative log likelihood. But it does also, GAPIT also estimates heritability of the trait. So in our case, it was 0.82, which is pretty high, which is what we expect for grain protein content in barley. And we can talk more about this in the actual live demo. So when it comes to analyzing your data, excuse me, so most of you probably don't have data in HAMPMap format. So what do? You can build your own HAMPMap file. If you have the genetic map and you have your genotype data, you can arbitrarily code your data as AA, AC, or CC to represent the different allelic states of your data, the SNP calls. That's one way to do it. Again, caution, GAPIT does not impute marker data for numerically formatted genotype files. So what do you do in that case? You can use another program like Tassel, FastPhase. There's one more out there that people commonly use. I can't remember it though. Or you can do the imputation in R. You can, if you have your data in numerical format, you can just take the average of the column and use that as for your missing data. So kind of whatever you want to do. If you supply it with numerically formatted genotype data, you have to provide the genetic map in a separate file. And then down here, finally, in the GAPIT function, the parameters will kind of change. Instead of a G, it changes to a GD to signify to GAPIT that it's a numerical type genotype file, and then also a GM for genetic map. And here's just a brief picture of code that you would use for running as numeric data. You can see the GD for numeric genotype, GM for genetic map, and everything else has stayed the same. And here's just a basic screenshot of what your genetic map should look like. You just need to have the name, the chromosome position of the markers. I did get asked the question, if you have a species that doesn't have the markers, what would you do? And I guess theoretically you could just make this all up. You just need to provide the file, and then your output would reflect that. Like if you just said chromosome 1, position 1 for everything, in the output file, that's how it would show up, as every marker would be at the same spot. And here's what numeric genotype data looks like in GAPIT. It's coded based on the 2-1-0 principle. Emma uses the same format. And so you have your taxa, your entries in this first column, and then each subsequent column is the name of the marker and the genotype call. And so that's what that looks like. And so problems, obviously consult the user manual, and they really have done a great job of providing code for just about nearly any situation that might arise. You can generate your own kinship matrix in another program. You can generate, your population Q file in another program, and it shows you how to do that, what type of code you need to use to import that data and run the analyses. So really, I can't stress that enough. This manual is pretty self-contained and really does a great job of explaining everything. Finally, check the website and check the data on the user manual that you're using. It gets updated as well as the code, and the updated manual reflects the changes in the code. So I know one time I was trying to use it and I couldn't even get the functions to load, and I just had to go back and realize that they had added some more stuff. And so once I incorporated that into my code, it ran perfectly. They do have tutorial and example data sets that I suggest maybe going through. They're pretty nice. They walk you through everything and some of the other features of GAPIT that I'm not going to cover today, like file splitting. And then finally, if you're just at wit's end and you're getting frustrated, you can always contact the makers. They're very friendly and extremely helpful. These guys know what they're doing, and they entertain most questions. So in terms of getting the most out of GAPIT, the code I showed you, we'll just do a basic analysis. This is a good starting point. But as you know, those probably aren't the results you want to use or the parameters you want to use either. So you have to do it a few times. You can, like I said, there's lots of stuff that you can adjust and change. And so I recommend doing that. Again, towards the end of the manual, they kind of give you some more of the advanced features like the model selection, which is nice. They provide more example script. And then finally, the one I gave you towards the end, there's some more advanced stuff that you can do. And we will cover that in a second. And briefly, I would just like to thank Alex Lipka and Ziwu Zhang for their help they've given me over the years, I should say. The extension team, Sean and John, thanks a lot, and the participants. And with that, I'm going to switch over and we'll actually run the GAPIT program. So this is what our studio looks like. Down here you can see, you can click on the Packages tab, and it lists all the packages that you have loaded in your libraries. And if you want to load one, all you have to do is click on, so like this BLR one, if you click on it, it just automatically loads it. You don't have to type any commit, like any code or anything. So we'll switch back over to the plot window. And then up here, provides you with some tools that you can use, like importing the data set, which I'll show you in a second. And then finally, it stores your workspace images over here. So this is that beginning code that we need to run in order to initialize GAPIT. And so we'll just highlight that, and you can click Run. And so, you'll sometimes get these warnings, and this is because I've ran it a few times before. We actually did the webinar, so this stuff's already loaded into R for me. And so then you would do the install packages. if you didn't already have these ones. And then finally, we'll use the library command to load all this stuff into our current session. And so looks like we're good to go with that. And then so down here we have the source code, and this web address obviously tells GAPIT where to find it. And then over here you see that it just loaded it all, and it's called the GAPIT function. So this way you know you have the program loaded. And then we'll run the code to get Emma, and it should show up. You can see that all these Emma functions have been loaded as well. So this is good. We know that we have all the programs loaded. We know that we have GAPIT loaded. So now we just need to get our data. So like I said, we can go up to Tools, do Import Dataset, and again, mine's always from a text file. And so you can go through and let's see. So if you wanted to select, you'd select numeric genotype file, and GAPIT will walk you through this. And it might have just, so I have to tell it whether or not you want a header on it, and how it's separated, and then tell it to import it. But we are just going to use the command line. And then, so that loaded both of our files. And you can see that because in our data, you have HAMPMap Geno has been loaded, as well as the phenotype data. So at this point, we have all our data loaded. And what's nice, if you want to look at it, you can just click on it. It'll open a new window up here. And so you can see our taxa and our proteins for each line. This is another great way to check to make sure things loaded right. To check the genotype data, again, we can see that's been imported correctly. We have our markers here, our chromosome position. This information is still NAs, and then we have our genotype data. So this is what's kind of nice about our studio is it allows a limited graphical user interface compared to just regular R. So again, we'll just kind of run through here and check the structure of our data. Again, you see here the data frame, the number of observations, the variables. So we know we have taxa and protein. That taxa is a factor, protein's numeric. It gives us those values. And then we'll just do a basic histogram. And again, the data looks really nice, good distribution. It doesn't look like there's any major problems. These lines of code, again, just will give us those summary statistics. And finally, I do like using this which is NA. So that way I know, I roughly have an idea of how much missing data I have before I even start analyzing it. And so this way I can just make sure that nothing got loaded funny and that we have the right number of lines. So in terms of changing the directory to do the actual analysis, you can go up here to Session and set Working Directory, and then choose Directory. At this point, you can navigate to wherever, you would just like click on this one, no compression, and hit Select Folder. And you can see down here in the console, it lets you know that it changed the working directory. So in this first analysis that we'll do, and that I explained in the presentation, we're not going to use compression. We're just going to do a basic analysis. And so you type in GAPIT. Again, it's case sensitive. Parentheses. Make sure you have commas after each line of code, and you can just highlight that and run it. And so our studio, what's nice is if it's working and thinking, it gives you the stop sign so that you know it's doing its thing. And so right here you can watch, and it just kind of keep an eye on it as it's doing the analysis. And while it's doing it, we can talk about not using compression. And so here's the code again for not using compression. Excuse me, for using it. This is the default settings. And so we've eliminated those lines of code. And like I said, GAPIT automatically tries to group things by 10. If you want to use a different parameter, like maybe you think 100's better, try and group lines by hundreds, you can change that. And so you would run this, And it takes a little bit longer because, it's trying to run the best analysis and, mush all these lines together. And so down here you'll see that GAPIT finished running. You'll see that there are 17 warning messages, but don't fret. If you actually look at the warnings, it's typically, it'll say like failure to remove some object. This hasn't impacted your analysis, and it should be fine. And so the output that you will get is quite substantial. And so you'll see if for just this one trait, it generated all these files. And I won't cover which each one is, but I guess that's why I caution you to make sure you change your directory so that you don't like put all this on your desktop or something. And then for comparison between compression and no compression, here are the top hits and their respective p-values. And so this is no compression used. We got these ones, we turned compression on, and our p-values got just a little bit smaller. In this one case, it got larger, which was kind of bizarre, but sometimes that happens. So that's just to give you a sense of, so maybe some initial analyses that you should try. If we go down, here's this third analysis, and I've tried to write up enough about it that it'll be explanatory. But pretty much, the input files are the same. The genotype data imputation is the same. But this kinship cluster and kinship group, there's some different options that you can use. And so one of these is complete and ward, and I know you might be scratching your head as to what those are. NERCs, if you consult the manual, it'll tell you all the various options that you can use for clustering and for grouping. And then finally, I threw in this model selection bit of code, and this uses the Bayesian information criteria to select the optimal number of principal components to use in the model. And so if you run this, it actually turns out that the best model that fit didn't actually use any of the principal components. It wasn't needed. And that's important because, population structure varies from trait to trait, and therefore you don't always maybe need to use the same number of principal components for each trait. And so by having this model selection on, it kind of makes it nice in that it does some of the thinking for you, and it tries to find the best model for your data. And when it does that, that optimal compression file that I was talking about, it'll list those settings. And then so here is the output that we got this time. And you can see that our p-values actually got, I would say a lot smaller. Not a lot, but smaller than they did when just using basic compression. So look at that number, and then we scroll up, and you can see that we made quite some improvement. And so that improvement might be greater, it might be less, depending on the trait that you're analyzing. So that's just one thing to consider when you're doing your analysis. And I can't stress it enough that the options of GAPIT are vast, and that it just takes some time to maybe try a few things out and see what works best for your data. This script file should have most information you need to run it, and don't think that you need to run RStudio to run all this. I've checked numerous times, and this code works just fine in regular R if you're more comfortable doing that. And so with that, that is all I have to say about GAPIT, and I'd be happy to take any questions.
Well, thank you very much, Duke. We really appreciated that, that review of GAPIT. And I just want to let everybody know that today's presentation, along with the sample data and the links to some of the outside resources, are available at the e-extension page associated with this. That e-extension page in 68355. So you can visit that, and the recordings of the edited recordings of this presentation will also be up at that site, probably by next week. And you can sign up for PBG News and get our newsletter at our PBG Works homepage. And you can also sign up for future webinars, view the archive, see what's coming up from extension page ending 60426. And I also wanted to remind everybody that prior to this webinar, you will be after this webinar, you'll be contacted via email with a survey, and the survey will be about this webinar, but also about the topics that you're interested in hearing about. And we take the survey evaluations quite seriously, and so we would appreciate if you would take the time to fill that out. So now I'm going to answer some of the questions that have come in. We've had a few, and if you'd like to add more, you can go ahead and type your questions in the question box on the GoToWebinar control panel. So the first question we got is, can you use 50K snips in this analysis?
You can use as many snips as you want. I mean, they analyze some of the corn data, which has, millions of snips. So it's not really limited to that. Again, if you're going to analyze that many snips, you would probably need to use a file splitting option in order to get all that data into R. But yes, as long as the data is formatted and snip like barley format, you should be good to go.
And we also got something that's really more of a comment than a question. One of the audience members was trying to access the Trudi ACA Toolbox, and it seems to not be working, and I've confirmed that. But I just want to remind everybody that the data associated directly with this webinar is available from the eExtension website, but the Trudi ACA Toolbox should be back online soon, so it's probably a temporary problem that they're having.
Sorry about that.
Do you have any comments on that, Duke?
I do not. I know the web developer, or excuse me, the content developer for that, and
might send him an email.
Yeah.
Another question is, what is the cutoff point for the FDR to declare significance?
Well, there's a lot of discussion about what that is. Personally, I've seen a lot of people use 0.05. That's kind of, depending on how many SNPs you're testing, you should take that into consideration. Sometimes, though, I feel most comfortable using 0.01. But again, I guess my answer would be that's kind of user dependent, and I can't give you a solid answer, but I would go with, at the bare minimum 0.05, and then to really just be sure, I'd use 0.01.
And so this question, I'll just read it word for word. GAPIT is performing single SNP test. Is it an efficient tool for GWAS analysis of several millions of SNPs according to multiple testing corrections?
So yes, GAPIT does test SNPs one at a time. Would it be appropriate for millions of SNPs, the FDR correction? Probably, because that FDR correction will take into account how many tests are being performed. And like I said, these guys, well, they work with the Buckler group, and the data sets they analyze are quite large, and they have more than, they do have millions of SNPs, and they're able to use this package to analyze that data, so they should be okay.
And the next question is, how can we set the level of significance on the Manhattan plot? On the QQ plot, sometimes the graph is below the slop and sometimes above the slop line. Why does that happen?
So with regard to the QQ plot, I think what they're meaning to say is the slope. So if it's too high above, that means you're declaring too many things significant. So basically, your results are, you're declaring too many false positives. If it's below the slope of the line, that means you don't really have enough power to detect significant associations. If I would have known this question was coming up, I could show you. In one case, I forgot to format the data properly, and I had, I think, like 500 missing values, and it was pretty much all those dots were on the x-axis. And first question, what was that, Sean? About?
The first question is, how do you set the significance level on the Manhattan plot?
That could be a little tricky. You would have to go into the source code, which is not too hard, and you can change that setting because it is based on, it calculates the significance value kind of for you. And so it'll change from analysis to analysis, but if you were really looking to do it, you would probably have to fiddle with the source code a little bit, which may seem daunting, but not totally unsurmountable. I couldn't give you a quick answer as to how you could do that just within the GAPIT function.
Okay, and the next question, and I think you may have addressed this a bit in your talk, is does it Is it required to have the chromosome position of each marker? How does GAPIT perform for crops like wheat, which don't have complete genetic maps?
So like I was saying, GAPIT just needs to see that the file's there. You don't necessarily, that information doesn't have to be correct. So I mean, you could make up your own genetic map just as long as when you interpret the results, you know that. And so, like I was saying, you could just create a genetic map file where everything is called chromosome 1, and the positions, you just label 1 through however many SNPs you have. And then GAPIT will analyze the data, but in the output, when it lists the the GWAS results, obviously you will know that the chromosome positions have no meaning. So I hope that answers the question. Yes, you don't actually need to know the physical or the genetic positions of these markers.
Okay, and so we have a few comments explaining why the TCAP website isn't working. The USDA servers are down because of the government shutdown. So when our government is working again, apparently then we'll have access to the TCAP data.
Okay.
And then we have a question, would you please include what Duke is saying in the final note to be released? So the recording of this webinar will be released, so his audio will show up. I think that answers your question. So when we post the recordings, there'll be the PDF versions of the files, but there'll also be the video. So you'll see him advancing the files and slides, and you'll be hearing his voice as well. And here's a question. Have you used GenAble before? Did you try to test the differences between the two packages regarding the results?
I have not used GenAble before. I can be completely honest. I use mainly Tassel, GAPIT, and I have done an analysis using SAS, but that was more painful than I'd like to admit. A lot of people say good things about GenAble, and certainly I don't think this is the only package that you should or could use to do this. I know there's another package called RRBLUP, which has a webinar on it, but I believe it's for the genomic selection function. But RRBLUP will also do association mapping. And so yeah, I can't tell you comparison-wise which one's faster, how the results will turn out. For GAPIT, since it uses compression and this P3D option, I typically will test my, run the same analysis in Tassel, because both Tassel and GAPIT use these functions just to confirm and make sure I'm doing everything right. But typically, I've checked my results with RRBLUP, which does not use these fancy options of compression, and the results are typically almost identical. I mean, the markers might change in terms of rank from like 2 to 3, and number 4 becomes number 1, but there's nothing too dramatic.
That is good to know. And I'm not sure if you'll know the answer to this last question, but someone wants to know if there have been scripts developed to deal with tetraploid SNP data.
Ooh, I do not. I'm sorry for that person.
And so another question is, have you seen any difference when you input kinship and PC data to GAPIT rather than generating it by GAPIT?
I have not, and that comes down, so I'm presuming maybe what this person's talking about is the different methods to calculate kinship. There's Van Raden, there's Emma, there's Louise, or Lucelle, I can't really pronounce it right. And so the kinship file, I haven't seen a big difference. And for most part, what I've read in the literature, Jeff Enlund has a paper about it, that in terms of doing GWAS, the method used to derive kinship, for the most part, doesn't have a large impact. And I don't want to, state that definitively, but if that person would like, I can give them the paper that they can reference. But no, I haven't seen a big difference, and I have not. So by virtue of that, I typically don't calculate my kinship outside of GAPIT, because I don't know, GAPIT will do it for you, and you can change the method that GAPIT uses.
Okay, well, I think that those are all of the questions that we've gotten, and thank you so much for joining us, and thank you so much, Duke, for the fantastic webinar.
Well, thank you, Sean.
Bye-bye.
Have a good day.
You too.